You know, in modern infectious disease management, the idea of one-size-fits-all dosing is, frankly, a relic. The real cutting edge is personalizing antimicrobial therapy for each and every patient. And in this explainer, we are going to dive deep into the cornerstone of that effort, Therapeutic Drug Monitoring, or TDM. We'll take it from the abstract theory right into the clinic.
This insight from Paracelsus back in the 16th century, it's not just some historical quote. It is the fundamental, day-to-day reality of antimicrobial therapy. That line between a dose that cures and a dose that causes real, irreversible harm, it can be razor thin. Our job is to walk that line with absolute precision.
So here's our game plan. We'll start with the core problem, what I call the dosing dilemma, and then look at its solution, TDM. From there, we'll get practical, see how it's used in the clinic, and then really put it to the test with a case study. We'll wrap up by looking at the bigger picture, why TDM is so crucial for antimicrobial stewardship.
Okay, let's jump right in with the dosing dilemma. You see, so many standard dosing regimens are built around this idea of an average patient. But here's the thing. In the real world of clinical practice, that average patient is a ghost. They just don't exist. Every single time we write a prescription for an antimicrobial, we are performing a high-wire balancing act. On one side, you've got efficacy. You absolutely need the drug concentration high enough to kill the bug. But on the other side, there's toxicity. You have to avoid drug levels that poison your patient, causing things like kidney failure or hearing loss. It is a constant, delicate trade-off.
And we actually have a term for this balancing act, the therapeutic index. It's that window between the minimum effective concentration, or MEC, and the minimum toxic concentration, the MTC. Now, for some of our most important drugs, think aminoglycosides, think vancomycin, this window is dangerously narrow. Go a little too low, the treatment fails. Go a little too high, and you risk serious, lasting harm. Trying to stay in that narrow window is so hard because every patient is a completely unique pharmacokinetic universe. Their kidney and liver function, their body composition, their other illnesses, even their genetic code. All of it dramatically affects how they handle a drug. It makes you think, why are patients not like lab rats? Well, because lab rats don't order pizza at 2 a.m., completely changing their drug metabolism. And, they tend to be a bit more cooperative. It's this incredible variability that explains why a standard dose can be perfect for one person, but toxic or totally ineffective for the person in the next bed.
So, if we agree that the average patient is a myth, how in the world do we dose for the individual? And that brings us right to the solution, achieving true precision through therapeutic drug monitoring. So what is TDM really? Well, the formal definition is the clinical practice of measuring specific drug concentrations in a patient's blood at specific times. But the goal, the whole point, is to actively manage that concentration, to keep it inside a preset therapeutic range. It's not just about drawing blood, it's a dynamic cycle of measuring, thinking, and adjusting. It is data-driven medicine at its best.
A really core principle here is that different antibiotic classes need different targets. For aminoglycosides, we're aiming for a high peak concentration relative to the bug's MIC, or minimum inhibitory concentration. For glycopeptides like vancomycin, it's all about the total exposure over time, what we call the area under the curve, or AUC. And for beta-lactams, it's about how long the drug level stays above that MIC. Each strategy is tailored specifically to how that drug actually kills bacteria.
But where does that target, the MIC, even come from? Well, it comes from the microbiology lab. They use standardized guidelines, like these ones from EUCAST, to define the breakpoints. This table tells us what makes an organism susceptible or resistant. So for an E. coli bug to be considered susceptible to gentamicin, its MIC has to be 2 milligrams per liter or less. That number from the lab gives us the concrete target we have to beat with our dosing.
Okay, let's pivot to the practical side of things. How does all of this theory actually come together in a hectic hospital ward? How do we take these complex ideas and use them right there at the bedside?
The TDM workflow is really just a continuous feedback loop. It kicks off when you prescribe and give the drug. Then, timing becomes everything. You have to collect that sample often at trough level just before the next dose at precisely the right moment. The lab gives you a number, You take that number and interpret it against your target range. Then you adjust, either the dose itself or the dosing interval. And this is key. You have to remonitor to make sure your change actually worked. Getting that perfect trough-level sample can be a real headache. It's the one time in medicine where being just in time is so much more important than being a hero.
So which drugs get this 5-star treatment? Well, it's the usual suspects, the ones with that narrow therapeutic window we talked about. Aminoglycosides like gentamicin, infamous for their kidney and ear toxicity. Glycopeptides like vancomycin, where we're trying to stave off kidney damage. Certain antifungals like voriconazole, which have crazy variable metabolism and can cause neurotoxicity. And more, we're using TDM for beta-lactams, especially in our sickest patients, whose bodies handle drugs in completely unpredictable ways.
Theory is one thing, but TDM's real power shines through in practice. So let's walk through a pretty high-stakes clinical case to see exactly how this data lets us make smart decisions when it really counts.
Okay, here's the setup. You're covering a 68-year-old patient with urosepsis who's on gentamicin. It's day 3 of the treatment. You notice the creatinine is climbing, up from 1.1 to 1.7. As you're thinking about that, the lab calls. The gentamicin trough level is 2.5 milligrams per liter. Your target is less than 1.0. So what do you do right now?
All right, let's break down the data. That high trough level is a blaring alarm bell. It tells you the drug isn't being cleared. It's building up. You combine that with the rising creatinine, and the picture becomes crystal clear. This is drug-induced kidney injury in progress. The clinical implication is unavoidable. The current dosing regimen is toxic, and you have to do something about it immediately.
So what's the action plan? First, you hold the next dose. Do not give it. Step 2, you extend the dosing interval. Maybe you go from every 24 hours to every 36 or even 48 hours to give the kidneys a chance to catch up and clear the drug. Third, you recalculate a new, smaller dose based on this patient's new, worse kidney function. And finally, you remonitor the levels before that next dose to prove that the trough has come down to a safe level.
And this case really gets to the heart of what makes TDM so elegant. The goal isn't just to slam on the brakes and stop the drug, which might put the patient's sepsis treatment at risk. No, the goal is to use the data to intelligently re-engineer the dosing regimen to steer the patient out of danger while still treating their infection effectively.
So, the definitive answer to our case question is this. You extend the dosing interval, you recalculate the dose, and then you watch like a hawk, remonitoring both the drug levels and their kidney function. This kind of active, precise management is exactly what TDM empowers us to do.
All right, for our last section, let's zoom all the way out. TDM is so much bigger than just optimizing care for a single patient. It is a profoundly important strategic tool in the global battle for antimicrobial stewardship. Some of these drugs have a therapeutic window so narrow it's more like a therapeutic keyhole, and TDM is what helps you find the door.
You can think of TDM's impact as having two sides. For the individual patient, it's all about safety and efficacy. It's about personalizing care to minimize harm and maximize the chance of a cure. But for public health, it's a game changer. By optimizing dosing, we reduce the selective pressure that fuels antibiotic resistance. It's a critical strategy for sparing our big gun antibiotics, like carbapenems.
And make no mistake, the need for this kind of stewardship is here and now. Look at this data from the Netherlands back in 2017. 8% of E. coli bloodstream infections were already caused by highly resistant microorganisms. This isn't some far-off threat. It is a clear and present danger that requires us to be much smarter about how we use the antibiotics we have left.
And this, right here, is the key stewardship takeaway. TDM lets us use our older, often narrower-spectrum drugs, like the aminoglycosides, with confidence. By using them safely and effectively, we can save our most precious, last-line agents for the fights where we truly have no other option.
So, I want to leave you with a final thought.
As the bugs we fight continue to evolve, and as our patients become more and more complex, can we really afford not to personalize dosing? And looking ahead, what's next? What role will things like AI and predictive modeling play in taking TDM to the next level? Because the story of personalized antimicrobial therapy is really just getting started.